University of Lethbridge and the Alberta RNA Research and Training Institute. I'd just like to take a moment to welcome everybody. Our university's Blackfoot name is Iniskum, meaning Sacred Buffalo Stone. The University of Lethbridge acknowledges and deeply appreciates the Blackfoot people's connection to their traditional territory. We, as people living and benefiting from Blackfoot Confederacy traditional territory, honor the traditions of people who have cared for this land since time immemorial. We recognize the diverse population of Aboriginal peoples who attend the University of Lethbridge and the contributions these Aboriginal peoples have made in shaping and strengthening the university community in the past, present, and in the future. And we're very grateful that you can all join us here today. And I'd like to invite our first speaker, Zachary Robinson, undergraduate student in the lab of Dr. Trushar Patel, part of the chemistry and biochemistry department here at the U of L. Zach, the floor is yours.
Awesome. Thanks, Laura. Going to go ahead and share my screen. Okay, amazing. So like Laura mentioned, my name is Zach. I'm an undergraduate student at the University of Lethbridge. And I've worked in the lab of Dr. Trushar Patel for about 3 years now. And so my talk today is on human DDX1 protein and its role in unwinding and retaining NEAT1 inside nuclear bodies. And so right away, the first thing I want to talk about is just a little bit of background on NEAT1 and why it's a relevant long non-coding RNA to study. And the main reason for that, or at least one of the main reasons for that, is its role in cancer. NEAT1 RNA has been identified as a biomarker in several different cancers. You can see a list down here. Where it's been shown to either correlate, the expression of the RNA has been shown to correlate with tumor progression, or the RNA has been shown to potentially have an active role in tumor progression, either as an enhancer or a suppressor. And this is just an example of the kind of data and the kind of studies that we'll be looking at for this kind of table, just generally showing that patients with higher levels of NEAT1 expression survive less. And so that kind of begs the question as to why that is. And so to try to understand that, you have to understand a little bit more about what NEAT1's role in the cell actually is. And the main process that NEAT1 is known to be involved in right now is the formation of these nuclear bodies called perispeckles. And so perispeckles are these bodies, as I mentioned, that consist of RNA and large chunks of RNA binding protein. And they're known to be heavily involved in gene regulation and gene regulation processes in cell. And NEAT1's major role in these bodies is to serve as a scaffold that facilitates the formation of the overall subnuclear bodies. With all the proteins will bind to NEAT1, and that binding will help to organize the proteins in such a way that the perispeckle can form. And so one of the really interesting things that some recent work has discovered about this process is that it actually may be regulated by specific RNA structures called G4 quadruplexes that have been identified throughout NEAT1. And because a lot of the proteins that are major components within these perispeckles have been shown to be G4 binding proteins in other unrelated work, the hypothesis is that it may be these G4 tertiary structures that recruit the proteins and facilitate the formation of these membreless organelles, the perispeckles. And so there has been some work done to kind of validate this approach. Some of the main proteins that the perispeckle consists of has been shown to bind specifically to G-quadruplex switch regions of NEAT1. And mapping of the NEAT1 long non-coding RNA has identified a large number of G4 quadruplex structures that are believed to exist based on computational tools and the tools that we have available currently. And so looking at all of this, we were really interested to investigate more the role of proteins in modulating G4 structures. There's been a lot of work showing that these structures may be the conserved motif that the perispeckle proteins bind to. And we wanted to look into what happens if a protein either facilitates the formation of more G4 structures in the RNA or unwinds G4 structures in the RNA. And that brought us to DDX1, which is a protein that has been shown in other studies to unwind G4 tertiary structure in other RNAs. And there had also been preliminary results that suggest that DDX1 may bind to NEAT1 in cellular. And so the goal for our study became to investigate whether DDX1 had a role in unwinding the G4 quadruplex structures that we believed were present in the NEAT1 long non-coding RNA. And so the first step in terms of trying to accomplish this goal was that we needed a way to produce both molecules and purify them through homogeneity. We were able to express and purify DDX1 using a standard E. coli expression and nickel affinity purification system. And you can see kind of the final results of that over here to the left. We chose a small quadruplex region of NEAT1 to use as a model. And because of that, we were able to produce this region using in vitro transcription assays and then purify using size exclusion chromatography to produce large amounts of this RNA as well. And so having accomplished that, the next step we took was to confirm that there were actual G-quadruplex tertiary structures forming in the RNA region that we selected. And so because we had based the region of RNA we chose off of computational methods, we wanted to experimentally validate that there were actually the G4 structures that we predicted in this RNA. And we did this two ways, using thioflavin T assays. We can see a much larger signal for our quadruplex switch RNA as opposed to the control, and also using circular dichrosal spectroscopy. We can see these extended peaks and dips at the specific wavelengths that they would be expected, had been characterized for this tertiary structure profile. And so at that point, we produced the protein, we produced the RNA. We've confirmed that the RNA does appear to form the structures that we believe it does. We then moved on to one final quality check where we used multi-angle light scattering, which gives you a value for the absolute molecular weight of a biomolecule in solution. And we used that to confirm that we were working with monomers as expected, which was important because then when we checked the binding between NEAT1 and DDX1 and confirmed that there was binding using an EMSA, we were then able to put the complex formed between the two species through multi-angle light scattering, gain an idea of what the size of the complex was. And because we know that DDX1 and NEAT1 are both monomers in solution, we were then able to determine the stoichiometry of the complex between the two appear to be a one-to-one binding ratio, which is interesting because there are several different quadruplexes in the RNA, which could potentially all recruit a different EDX1. And so after gaining this preliminary information, the next thing we wanted to do was to gain more structural data on the complex, and hopefully use that to get a little bit of a better idea on how it works. And so we did that primarily using small angle X-ray scattering. And so in this technique, you can look at the pattern at which your sample scatters X-rays, and there's some structural information in this data that you can exploit to try to generate a low resolution structure and get an idea what your protein, RNA, and complex look like. And so on this slide, I've included two different plots that kind of summarize the X-ray scattering data we were able to collect. To the left, you have a Guinea plot, which is more of a quality control measure. And basically what you're looking for here is just to see how well a linear regression fits the points and the data. And based on that, you're just looking to see if you see any, especially curvature in the upper left corner, and that may suggest that your sample is aggregated and that your data is not really that reliable. And so after performing a quality check on the data, we then moved ahead to generate the paired electron distribution function, which essentially tells you how many pairs of electrons are each distance away from each other throughout the molecule. And so this is where most of your structural information is. And from here, you can generate ab initio models that would create paired electron distribution functions that fit well with the experimental data that you've collected. And so having done some basic quality checks, we moved ahead with generating those ab initio models. And we were able to generate a low resolution structure for MEAT1 as well as the DDX1. And then on top of that, because we have data for each individual component as well as the complex, we were also able to use a program called MONSA to generate a prediction of what the complex would look like, taking into account the scattering data for the complex, as well as the scattering data for each of the individual components when they're on their own. And so what you can see here is the data after we clusterized all the different models that we've generated based on similarity. And I've displayed the representative models for each of these clusters up here. And so I guess some of the main features that I would draw your attention to is number one, we've learned that this isn't an end-to-end interaction. It looks like DDX1 appears to dock kind of to the side of the RNA, and the RNA wraps around, which makes sense given the relative D masses that we were able to determine from P-value plots. And then also interestingly, it looks like there may be a couple different positions and sites for DDX1 to bind the RNA, which would be consistent with the fact that there's 4 to 5 different potential quadruplex structures throughout the RNA that we hypothesized that DDX1 may have a role in unwinding. And we can even, though we need high resolution data to investigate further, even speculate that potentially DDX1 has this processivity. We'll go along the RNA and unwind each quadruplex subsequently. And so after we had confirmed that the actual interaction occurs, we've confirmed the presence of the G4 quadruplexes in the RNA, and we've structurally characterized the interaction, albeit to a low resolution. The next step that we wanted to take was to try to prove functionally that DDX1 does unwind MEAT1. And we did that using these helicase assays. And what you do for these is you design a DNA probe that is complementary to the RNA structure that you believe is being unwound. And theoretically, if you have the structure, the RNA should be unable to base pair to your probe, and you should see no binding. Whereas if you add a helicase that may potentially unwind these tertiary structures, the RNA should now be linear, and that should be available for your probe to base pair, and you should see binding. And so that's how we tested it. And you can see the results over here to the right. We designed a probe for the 4 sequences that we were pretty sure there was a quadruplex at, as well as the one where we were a little more uncertain based on what the computational numbers and the output we were getting. And we found that in every case, there's highly significant difference between the binding of the probe to the RNA without DDX1 and the binding of the probe to the RNA with DDX1 present. And this supports our hypothesis that we do actually see unwinding of these regions of the RNA by DDX1. And so at that point, we've completed a lot of the in vitro experiments we wanted to do, and we moved on to trying to further characterize how this interaction works in the cell. And for that, we began by using pull-down assays. And so to complete this, there's kind of a little graphic down here. I purified DDX1, immobilized it by the His tag on magnetic beads, and then washed the beads with the immobilized DDX1 with a cell lysate. At that point, we then washed the beads quite vigorously to remove anything that wasn't tightly bound to DDX1. And then we performed a couple experiments using the extract that we got from the beads following washes. And so the first experiment we did was to try to investigate whether there was more DDX1 being pulled down, or more neat one, so being pulled down by DDX1 or by a control protein, to try to investigate whether this interaction still occurs in an environment that much more closely simulates a cell, with all the other proteins and RNAs and components also floating around there in the tube, as opposed to just DDX1 and just one. And what we saw was we did see a significant increase in the amount of NEAT1 that was present in the samples obtained and pulled down using DDX1, as opposed to the amount that was pulled down using green fluorescent protein. And so that helped to further validate that this interaction was something that potentially still occurs in the cell and isn't an artifact of our in vitro system. There we go. Okay. And so the next step that we took after this is we still had our pull-down fractions, and we wanted to investigate what the protein interactome of DDX1 looks like, because that would enable us to compare this to the protein interactome of NEAT1 and see to what extent they're similar. And so we're still working on this a little bit, going through the data. However, the first thing we did was to just kind of classify all the proteins that we were getting as part of the DDX1 interactome, more or less as a validation of the assay. And what we saw was a lot of the kind of protein roles that were enriched to a high-fold enrichment with processes that DDX1 was known to be already involved in, such as mismatch repair. And so that was nice because it allowed us to some extent confirm that we were pulling down proteins that we already knew DDX1 interacted with, which was a good sign. After that, the next step we took was to look at many of the known heterospeckle proteins and see if those binders were also showing up in the DDX1 protein interactome. And the two that I'd really like to draw your attention to are nono and phos. These are both heterospeckle components that we identify in our DDX1 pulldowns. And interestingly, they're also known to bind to G4 quadruplex tertiary structure in RNA as well. And so after this, the next thing we wanted to investigate is, I guess, how DDX1 affects these proteins, and specifically how the absence of DDX1 would affect these proteins. And so we knocked out DDX1 in the cells, and then measured how what the change would be to the expression of RNA coding for NEAT1, and then also several important heterospeckle proteins that have already been shown to be a major component. And what we saw was that NEAT1, NONO, and heterospeckle protein 1 all saw a significant increase in the amount of transcription that was occurring following DDX1 knockdown. And so that was interesting to us. Kind of our first instinct was to hypothesize that DDX1 knockdown would then lead to greater amounts of heterospeckle formation because we are seeing greater amounts of all of these components being transcribed when we knock down DDX1. However, to investigate this further, we use fluorescence in situ hybridization along with confocal microscopy to try to observe DDX1 and NEAT1 in the cell and try to see potentially what effect DDX1 knockdown had on NEAT1 foci, on heterospeckle formation. And that's the data that I have over here. And so you have two conditions here, the wild type cells and the cells with DDX1 knocked down. And we use the fluorescence in situ hybridization to try to visualize where NEAT1 is in these cells, as well as just anti-DDX1 antibodies to try to visualize where DDX1 is in the cells. And one of the first things that you notice is that there's these kind of foci that have a higher NEAT1 concentration than anywhere else in the cell. And it appears that DDX1 is also localized in these regions of high NEAT1 concentration. So it was, first of all, really interesting to see that there potentially is some colocalization between these two molecules. And then even more interestingly, when DDX1 was knocked down in the cells, we saw that there was essentially the foci, the NEAT1 foci disappeared, and the RNA became more dispersed, which was kind of not what we expected based on our hypothesis from the last slide. And so this is something that we're still working on. However, the main hypothesis that we have for this to kind of reconcile these two pieces of data is that DDX1 may potentially have a role in modulating the structure of NEAT1 as previously seen, and that modulation of the structure may potentially be necessary in order for nuclear bodies such as a perispeckled form, or alternatively, just for this localization of NEAT1 in these bodies to occur. And that's one of the things that we're looking to investigate further, potentially in the future. So I guess kind of just to summarize the results for this project, we confirmed the presence of G quadruplex tertiary structure at the positions that we predicted it would be. We confirmed that there is a binding interaction between human DDX1 and NEAT1 long non-coding RNA. And we did confirm that DDX1 had some kind of unwinding effect against NEAT1. We also observed the colocalization of these two species within the nucleus and within specific compartments in the nucleus. And we saw that when GDX1 is knocked down, these compartments are no longer observed. I think one of the big things we're looking to do in the future is to try to gain a high resolution structure of the complex to try to better understand what the mechanism for unwinding is. And we're hoping to do that with cryo-EM. We're also hoping to follow this up with a couple more confocal microscopy experiments, tracking the position of proteins like FUS and other known perispecial proteins, and seeing what effect GDX1 knockdown has on their behavior. And so I think lastly, I just want to say a really big thank you to my in-lab mentor, Dr. Ivo, as well as my supervisor, Dr. Trushar Patel, as well as everybody who's given me a hand with this project. And I think if there are any questions, I can take those now.
Awesome. Thank you so much, Zach. Great job. And yes, questions are open. You can use the Q&A feature that is available at the bottom of your Zoom screen. So feel free to ask any questions of Zach. I think we have about 5 minutes on the clock until our next speaker. You said in your closing slide that you are actually going to do cryo-EM. What's your plan for that? Have you started that process yet?
Yeah, so we got some really exciting news on that front just a couple of days ago. We applied for some microscope time at the F2C2 facility at Stanford. And just in the last week, we were able to get our samples on the microscope, and they were able to collect some data for GDX1 on its own. So we're really excited to begin processing that data, and also trying to, in the future, collect some data for the complex, as well as potentially meet one on its own.
Awesome, that is really exciting. I have another question. So just going back to your MULS data with the one-to-one stoichiometry. So it's processively binding and then unwinding these G quadruplexes. Is it binding the same one each time, or is it binding different ones? Like, is there a preference for which one it binds first? Do you guys?
Yeah, and so we really don't know a great answer to that one. What we saw on the helicase assays is that kind of all 5 of those regions are unwound. And it does look like from the SACS data that you potentially have it binding to different regions. I think it would be really difficult to confirm that we are seeing processivity versus the helicase binding to each individual quadruplex and then letting go. But we're hoping that cryo-EM might be able to give us a little more information on that. If we see a whole bunch of different kind of conformations for the complex, we could potentially try to classify all of those differently and build some kind of structure for each of them. Though that is a little ways off, and we need to get a better idea how the complex performs on grids and behaves on grids.
Okay, thanks. Well, I have so Tom Schneider is asking, is ATP or GTP used for the unwinding? And in either case, can you measure how many G quadruplexes are unwound per nucleotide?
Yeah, okay. So I guess for the first question, this one was interesting in that the literature actually shows that ADP may be necessary for the unwinding. And so that is what we added to facilitate that. Could we measure how many G quadruplexes are unwound per nucleotide? I'm not sure if that's something that's super feasible now. One experiment that I would be interested in trying over time would be to add unlabeled oligos just to try to cap and prevent G quadruplexes from reforming. And then we might be able to, I don't know if we'd be able to get a specific number, but we might be able to quantify the rate at which DDX1 is unwinding. Because the issue we have trying to address that question right now is that if you just let DDX1 unwind and then try to measure if there's less quadruplexes in the sample or not, is that the quadruplexes can just reform once DDX1's gone. But we're hoping that potentially if we design an oligo to base pair and prevent that quadruplex from refolding because it's double-stranded now, that we might be able to use something like thioflavin T to quantify, I guess, the rate at which the unwinding is happening. And we could potentially modulate which probes we use to get an idea of if it's unwinding different quadruplexes at different rates. So I'm not sure if we'd be able to generate a specific number like that. Sorry, I hope that.
No, wait, that's okay. We're short on time, so I'm gonna stop you there. But Patricio has a question as well. You can answer in the Q&A directly to them if you would like to do that while we have our next speakers. So I'd like to invite Dr. Indu Negi, also from the University of Lethbridge and the Alberta RNA Research and Training Institute, to give her presentation. Whenever you're ready.
Thank you, Laura, for the introduction. Let me share my screen. I hope my presentation is visible to you all.
You're good. Good.
Yeah, so, okay. So hello, everyone. I'm so happy to be here and to talk to you about the work that we are doing on unraveling the dynamics of ykkC-I riboswitch, insights into bacterial metabolism and metal ion-induced allostery through molecular dynamic simulations. So first of all, what are these riboswitches? These are non-coding RNAs which are located at the 5' untranslated region. They sense different metabolites based on their concentration levels. Now, these riboswitches, they constitute two domains, the aptamer domain and the expression platform. This is the schematic illustration to show how these riboswitches work. Now, the aptamer domain, it binds with ligand, and this binding, it brings some conformational changes, and this makes the expression platform available for the genes expression. Now, it's based on the conformational, how these conformational changes happening, it either switches on gene expression or switches off gene expression. Now, these riboswitches, they work or they act at either transcriptional level or translation level. So it's like molecular sensors which are functioning based on the concentration of, based on the changes in the concentration of ligands. So in past 2 decades, like more than 55 riboswitches have been experimentally validated, and these riboswitches, they sense diverse range of biologically relevant metabolites are known as ligands. And these ligands are cofactors, derivatives that are derived from RNA, RNA precursors, signaling molecules, elemental ions, amino acids, and different other metabolites. Now, these riboswitches, they have diverse structure in their aptamer domains. And this diverse structure allows them to bind to their respective cognate ligands with high precision and specificity. So now what's interesting is that this, it's like the single riboswitch can not only detect only like single ligand, but they have capability of detecting more than one ligand. However, there are also tandem riboswitches, which are like, they are more than 2 or more riboswitch, they are found together, and that can sense either the same ligand or different ones. Now, our research, it is centered around guanine riboswitches. Now, these riboswitches, for like For long, they have been considered as orphan riboswitches. These riboswitches, the structure of these riboswitches were elucidated, but the specific ligand that they bind remain unidentified. So, like recently in 2016, the ligand for these riboswitches were revealed, and it was revealed as guanidinium cation. These guanidinium cation, these are protonated form of guanidine. Now, like uh, these guanidine, they are chemotropic agent. Means like when the concentration of these guanidine increases, it interacts with the tertiary protein structure of bacteria and tends to denature complex protein structure. And they are also considered as toxic for bacterial cells at high concentrations. Now, to combat this toxicity, bacteria have developed mechanism where they control gene expression to deal with this toxicity. And the gene expression is controlled by these ykkC family of riboswitches. Okay, first let me, yeah, these ykkC family of riboswitches. Now these genes express, these genes control the expression of these enzymes, carboxylases, deaminases, hydrolases, exporters and transporters. These enzymes, they detoxify these toxic guanidine and convert it into less harmful compounds. Or somewhere these exporters and transporters, they absorb these guanidine ions, and they export these toxic compounds out of the bacterial cells. Now, binding, understanding the mechanism of how these binding occurs can help in understanding the different therapeutics that can be possible to deal with these bacterial infections. So now in literature, or like experimentally, 4 types of these ykkC family riboswitches have been identified. And these G1 and G4 riboswitch, they act at transcriptional level of gene expression. However, the guanidine 2 and guanidine 4, sorry, guanidine 2 and guanidine 3, they act at translation level of gene expression. Now, when the guanidinium cation, it interacts with the atom domain of these riboswitches, there is some tertiary interaction. Now, this tertiary interaction is loop-loop kind of interaction in guanidine 2 and 4 riboswitch. However, it is side-by-side packing in guanidine 1 and formation of H-type pseudoknot in guanidine 3. Now, now these are the details of crystal structures that have been reported for these riboswitches. However, for guanidine 4, the crystal structure have not yet been reported. Our current work will focus on understanding the guanidine structural dynamics of these guanidine 1 riboswitches, and we'll use computational methodologies to understand this guanidine 1 riboswitch, how this guanidine is controlling the gene expression. So why are we using these computational methodologies? Now, understanding the dynamic insight into these complex biological systems using experimental techniques is quite expensive in terms of building those laboratory lab equipments and materials. So in contrast, the computational studies, they provide or they offer cost-effective and rapid exploration of these structural dynamics, which generates data that can be used for predictions and building hypotheses, which can help experimental researchers to prioritize their experiment, focusing on the most promising areas. So now, using those, our computational analysis, we'll try to understand the structural dynamics. And understanding those structural dynamics of G1 riboswitch domain may have implication in providing insights of guanidine 1 riboswitch controlling its gene expression regulation. Further, these insights will facilitate the development of more efficient biotechnological applications or tools like biosensors, and can aid in the design of these riboswitch targeting drug developments. So to understand these to understand the guanidine-1 riboswitch gene regulation, we first focus on the crystal structure of guanidine-1 riboswitch. Now, the green color represents the 5T83 crystal structure, and the blue represents 5U3G, and the red color represents the 7-MLW crystal structure. These are the ions which were resolved around the binding pocket area for these three crystal structures. Now, when we superimpose these structures, we observe that the overall global structure of these riboswitches looked similar. However, there was variability in the loop regions. These are the loop regions. So further, when we looked into the detail of the crystal structure, we observed that the conservation of these nucleotides, which are forming these helical structure, was proposed to be more conserved in case of this green crystal structure, which is 5T83. Now, additionally, there were also observations which suggest that there are minor structural variation in the green crystal structure, and such minor variation may play an important role in RNA folding, and so RNA folding and aptamer stability. So we took this crystal structure for our further analysis, and also to understand the effect of ions on these aptamer domains, we took the crystallographic position of potassium and magnesium ion from 7-MLW crystal structure. So first thing is to build these models for our computational studies. We did molecular dynamic simulations, and we built 4 different models. The first one is free model, where we don't have any guanidinium ion or any crystallographic ions in the binding pocket area. And the second model is where we took aptamer domain, and there is guanidinium ion present in the binding pocket area, and it doesn't have the potassium and magnesium ions. Further, we took the third model. In third model, we just considered the ions around the binding pocket area. And in the fourth model, we took both guanidinium cation and crystallographic ions in the binding pocket area. And further, all 4 systems were neutralized using potassium ions, and we performed 2 microsecond molecular dynamic simulations for each system, and each system was replicated for 3 times. Now, before diving into the analysis, what we observed and what results we found, I would like to discuss the overall structure of guanidine-1 riboswitches. Now, these guanidine-1 riboswitches, they are composed of different helical structure. The first one is P1, followed by P1A, followed by P1B. This P1B is the loop region, then P2 loop region, and which is followed by this helical stem, that is P3 region. Now, this is the this is the ligand binding pocket area, which is formed between the P1A helical region and P3 region, and this is the structure of our cognate ligand, guanidinium cation. And these are the residues which are directly participating in non-covalent interaction with the ligand, with the guanidinium cation. This is the 2D representation of guanidine-1 riboswitch, which clearly shows how beautifully these helical structure of P1A are coaxially stacked over each other. This is the P2 loop region followed by P3. Further, from our experimental studies, not our, but from the literature, we observed that these residues of P3 helix, they play an important role in forming the complex, in stabilizing this complex aptamer domain when there is ligand binding. And hence, this aptamer domain on binding with ligand affects this expression platform, which is attached to the P3 helical region. Now, on binding with ligand, this is how our helical structure of guanidine-1 riboswitch looks like. So, first, we'll discuss about the overall global structure that we obtained from our molecular dynamic simulations, and we've tried to analyze the structural fluctuation of these aptamer residues. Now, I have used these color bars to represent different helical regions over here. So these are the P1 residues, and similarly it goes like that. Now we observe that on binding of ligand and ion, on binding of aptamer domain with ligand and crystallographic ions, the fluctuation of residues, it decreases. This is the green color represents ligand ion bound state. However, this the behavior of this fluctuation was similar to the aptamer domain when it was bind with the crystallographic ions. So this is the P1A and P2 region. However, when we looked at the P3 region, similar trends were observed. The ligand ion bound state was least fluctuated, and the free aptamer state was highly fluctuated. And further, since this there is stable, we can see the fluctuation decreases. Hence, it can affect the expression platform that is attached after this P3 helical region. Now, when these biological systems are in simulation, they are dynamically moving all around the box area. Now the structural conformational dynamics play vital role in function of many riboswitches. So we try to extract the most dominant motion of these aptamer domain in different scenarios. So in the free state and ion-bound state and ligand-bound state, we observe that these P3 helical region and P1 helical region tends to move against each other. However, when we compare it with the aptamer domain containing both ligand and ion-bound ions of crystallographic ions, we observe that these helical regions P1 and P3 helical region tends to move towards each other. So when we look deep into the dynamical motion, we observe that this opening is highly influenced by the stacking of residue at G40 at 40 position, that is G40 and A84. And when we look into it, we observe that the stacking, it tends to disrupt as the simulation duration increases, thus suggesting that there is this stacking may play an important role in maintaining the stability of aptamer domain. So when we looked into this stacking occupancy of this G40 G84 pair, we observed that this stacking interaction was highly occupied in case of aptamer domain with ligand and crystallographic ions in binding pocket area. And for free state and ion-bound state, this stacking was least occupied, thus suggesting the importance of this G40 and G84 stacking in stabilizing aptamer domain. Further, we looked into the second most dominant motion, and we observed that there was this A63 nucleotide was observed to be highly flexible in all aptamer domains. But this flexibility was observed to be high in case of aptamer domain without ligand and ion and with ligand system. However, when we compare it with aptamer domain consisting of crystallographic ions and both ligand and ion, we observed that this A63 nucleotide, it wasn't that flexible. It was stabilized by some non-covalent interaction. So to look into this non-covalent interaction, we try to analyze the stacking interaction of A63 with the nucleobase that was opposite, which was trying to stack with A63. And we observed that in all the four cases, the stacking occupancy of these nucleotides remained same. That is, it was occupied for 40% throughout the course of simulations. So now when we looked at the motion, overall motion of the aptamer domain, we tried to look into the base pairing interaction that are formed and which play an important role in stabilizing aptamer domain. And we saw that all the Watson-Crick-like and non-Watson-Crick-like hydrogen base pairing remain 90% occupied throughout the course of simulation. However, when we look deeper into these base pairing interaction, we found a few of the key hydrogen bonding interactions which were stabilizing the helical region. This is the hydrogen bonding interactions which were stabilizing the P3 region, and this region is near to the ligand binding pocket area. And this hydrogen bonding interaction was observed to be highly occupied. Not highly, but yeah, in case of ion crystal bound state and ligand ion crystal bound state, this occupancy was increased by 10% compared to the other two states. Further, we looked into the hydrogen bonding interaction that was stabilizing this P1A helix and P3 helix on binding near to the ligand binding pocket area. And we observed that this hydrogen bonding interaction was further stabilized when there was presence of crystallographic ion, or when there is both ligand and crystallographic ions are present in the binding pocket area. So next we look into the a quadruple kind of hydrogen bonding interaction that was playing important role in stabilizing this P1A and P3 interaction. And we observed that the interaction of G40 and A65, this is for P1A and this is P2, it was increased in case of aptamer domain with crystallographic ions, and in case of ligand and crystallographic ions, suggesting that when there is when there is crystallographic ions, these hydrogen bonding are highly occupied throughout the course of simulations. But however, when there is both ligand and crystallographic ion, these crystallographic ions are playing an important role in stabilizing these hydrogen bonding interaction, and thus stabilizing the ligand binding pocket area. Further, we looked into the overall correlated motions between distant region of aptamer domain, and we observed that so these legends over here, when the intensity of red color is high, it means that there is these motions between these residues are highly correlated. And when the intensity of this blue color is high, it means these residues are highly, and the motion of these residues are highly anti-correlated. And the the white color represents no correlation at all. So when we look at the correlation motion, and we observe that there was increase in correlated motion between the P3 and P2 region with P1 and P1A helical regions. And this correlation increases as we move from free state to ligand bound state, to crystallographic ion bound state, and to ligand and crystallographic ion bound state. Further, the correlated motion was also observed to be increased between the P3 region with P1A and P1. And this also followed the same trend, and this intensity was high in case of ligand ion bound state. Further, in the third correlation that we observed was decrease in anti-correlated motion between P1 and P1A. And this decrease in anti-correlation was highly enhanced in case of ligand ion crystal bound state. So further, when after analyzing this correlated motion, we zoomed in and we looked into the compactness of ligand binding pocket. And we observed that the ligand binding pocket in case of open state, so in case of free aptamer state, it was highly flexible. It was attaining 2 regions of radius of gyration. Further, when we compared these compactness of ligand binding state, we observed that the probability of compactness in case of ligand ion bound state was observed to increase to modest degree compared to the aptamer domain with crystallographic ions. And however, when we compared it with ligand bound state, the probability was observed to be highly increased. The compactness observed to be highly increased. So after this understanding the compactness of ligand binding pocket, we looked into the interaction of these guanidinium cation with the residues of aptamer domain. And we found that compared to the ligand-bound state, the aptamer domain with ligand and crystallographic ion, the occupancy of hydrogen bonding interaction increases, and thus suggesting there is some role that is being played by these crystal ion in increasing the compactness of ligand binding pocket. Thus, it increases the hydrogen bonding occupancies between the cation and the residues. Further, understanding about when we looked from our overall structure, overall analysis, we see that there was some role that is being played by these ions. So we looked into the binding position of magnesium ion around the ligand binding pocket area. This is the pattern of magnesium ion binding that we took from crystal structure, and we see that these magnesium ion are coordinating with the phosphate oxygen of residues that form part of P3 helix and P1A. When we compared it with our structure, we found that the magnesium in our system, they occupied this position for more than 90% of for more than 90% of the simulation course. Further, when we try to analyze the distribution of these potassium ion around the binding pocket area, we observe that, so this is the crystal structure which shows how the potassium ion interacts with the different residues. So when we analyze our potassium ion binding distribution, we observe that in the free bound state, these potassium ions were all over scattered in binding pocket area. However, in case of ion crystal bound state area, the density of potassium, it tends to move towards the position where ligand is present, but still it is near to the binding pocket area. Further, in case of ligand-bound state, the potassium density distribution were observed around these key nucleotides, the key residues. And in case of ligand ion-bound state, the density of potassium ion can be seen clustered around these A39 and C80 nucleotide, and suggesting that there is some coordination that is happening between A39 G80. So bringing all these analyses together, and we observe that these correlated motion of P3 region is enhanced with P1A, with P1 or other helical region of aptamer domain in the presence of ligand and crystallographic ions. This is attributed to the increased occupancy of hydrogen bonding between these base pairs. That forms part of P3 and P1A helical regions. Thus, suggesting, and however, these hydrogen bonding interactions were observed to be highly occupied in case of ion-bound state as well, which suggests that suggesting that there is some role being played by these crystallographic ions. Further, the stacking interaction of G40 and G84, it was also observed to be increased in case of both in case of ligand ion bound state. However, this stacking was also occupied in case of ligand bound state, which ligand bound state, but enhanced in case of aptamer domain with both ligand and ion bound state. Thus, it suggests that the crystallographic ions, they are playing role in pre-stabilizing the binding pocket area. And further, the crystallographic ion, it creates a stable environment for ligand binding, and which suggests that this enhancement in key interaction between ligand and the residues of riboswitches is influencing the presence of these crystallographic ions are influencing the conformational dynamics, which is essential for ligand for high or stronger interaction of ligand with the residues. So thus from our all analysis, we it was observed that there is some cooperative interaction that is happening between these ions and with the ligand, and this cooperative interaction are like finely tuned, and this fine tuning is enabling these enables the specific recognition of guanidinium cation by these guanidine 1 riboswitches, and thus affecting the regulatory function of gene expressions. So with that, I would like to thank Professor Wetmore for letting me do this research in her lab as a postdoc fellow. I'm grateful to my lab members for their constant support and feedback, and big thanks to our funding agencies and Digital Alliance for giving us the research, computational research sources that we need for our computational work. Thank you so much, and I'm ready for your questions.
Thanks, Indu. That was great. Unfortunately, we are out of time. I'll see if we have. Tom Schneider is asking, has the binding energy or KD of guanidinium to the aptamers been measured?
Yeah, like I have just discussed about the occupancies of these interaction. About the energetics, we are still working on those things.
Awesome.
Yeah.
And yeah, I'd encourage anybody who has any other questions to reach out. Both Zach's and Indu's contact information can be found online, I'm sure. And if you're interested, reach out to anyone from the Alberta RNA Research and Training Institute. Thank you all for coming. I really appreciate you spending some of your afternoon with us today. Have a great rest of your day. Thank you.
Thank you.